in late 1960s. So these are the major technical milestones in sequencing technology. You don't need to remember all of them, just remember some of them, starting when they first happened, and we are going to talk about several of the major technologies. And the first generation sequencing technology, also called Sanger sequencing. It was named after Frederick Sanger. And before I go through that, so this is a list of genome application milestones for sequencing technology. So once a sequencing technology, as the sequencing technology was developing, it was also applied to sequence different genomes, and it was applied to not only sequence DNA, but also different kinds of DNA from different sources, like RNA, and also different kind of DNA sequencing. So for the genome, we know that from the Human Genomes Project, the first human reference genome was available in 2001, and it's interesting. I just want to bring to your attention that in about 2010 and 2012, we get the genome for two archaic humans. They're kind of like, they are a sister group of modern humans, and they are already extinct. But we are able to extract DNA from their, it's not really fossil, but it's from their ancient remains, and we are able to recover their DNA information. And it's very interesting that for Denisovans, later in our class, we are going to devote one lecture to archaic human. For Neanderthal, from archaeological study, we have skeletons for Neanderthal. For Denisovans, they only have a finger, have a tiny bone. We don't actually have a skeleton for Neanderthal. But based on the DNA from that finger bone, they were able to identify a new human subspecies. So it was only made possible by the sequencing technology. We're going to talk about that not today, but in one lecture later. And these are different applications of sequencing. I highlight all of them because in this lecture, I'm going to talk about most of these applications. And at least for those that I talk about, you need to know what they do. Fred Sanger, Sanger sequencing was named after him, and he was the first person to do protein sequencing. In 1953, it was the sequence of the bovine insulin. And so he was awarded a Nobel Prize in 1958, it was very soon. And then he moved on to work on RNA sequencing. So in 1967, he determined the sequence of this ribosomal RNA from E. coli. And if you go back to look at when did the first sequencing of RNA happen, it was 1965. So Sanger was only 2 years after that. So he is really a pioneer also in RNA sequencing. And then in 1977, Sanger and Khalid developed a new method to sequence DNA. It's called chain termination method. And this is also called the Sanger sequencing method. It becomes the standard sequencing tools in the early days. So Sanger received another Nobel Prize for working on DNA sequencing. So he is one of the few people that received 2 Nobel Prizes. So the first generation sequencing technology is also called Sanger sequencing. To do a Sanger sequencing, you need to have, so this is the template. Template is your target DNA. You want to determine the sequence of nucleotide on that target DNA. In order to do that, you need to have a primer, and then you need to have a DNTP, deoxynucleotide. Basically, those are the building blocks of DNA. So if you have primer, you have DNTP, and you have a template, you can synthesize the complete complementary strand of that DNA. And the key feature of this technology is that they need to have DDNTP. that's dideoxynucleotide. And this nucleotide has very similar structure to DNTP, but the thing is that they don't have the 3' hydroxyl group. So they can be incorporated in this process in the DNA synthesis. But once they're incorporated, they won't be able to accept another DNTP. So the synthesis will terminate once the DNTP is incorporated. The things that you can control, so you need in the solution, you need to have DNTP, you need to have DDNTP. The trick here is that you need to control the concentration of DDNTP so that they're at a very low concentration. So only the probability of a DDNTP at this position is relatively small. Otherwise, if we have a high concentration of DDNTP, then everything will be DDNTP. It won't elongate. So you control the concentration so that DDNTP will be randomly inserted at some position. So once it's inserted, it will terminate. So you have a bunch of product from that DNA synthesis process. They are of different length and they terminate at a specific DDNTP. And that DDNTP is fluorescent labeled. And then you have this bunch of fragments of different length. You run them through a gel. And then if you run DNA through a gel, we can separate them based on different length. So we can determine the length of that DNA fragment. And then use fluorescent, you can determine which nucleotide is at that specific position. So you run through the gel at different position. So this is the chain termination reaction. So the incorporation of DDNTP terminate the synthesis at specific position. And then you run through this gel and you get this figure read by the computer. A better illustration is this. So this is kind of like you run a gel, and then at each position, based on the fluorescence, you can determine what kind of nucleotide is at this position. And this is the output of a typical Sanger sequencing. It's called chromatograph. And the major features of the Sanger sequencing is that you need to run the gel to separate different DNA fragments. So you need to rely on electrophoresis. And Sanger sequencing has very high quality. The error rate is relatively low. And right now, Sanger sequencing is considered as the gold standard of sequencing. If you use other sequencing technology and get some result, you want to confirm if that result is accurate or not. What you need to do is that you design a Sanger sequencing to confirm that. And the read length is about 800 base pair. Basically, you can read 800 base pairs, and that's considered as relatively long. Later, when you compare to the second generation sequencing technology, you can see that the 800 base pair is actually quite long. And so this is the typical result from a Sanger sequencing one. And usually at the beginning, the quality is relatively bad. That's because of the primer binds nearby. And if we go along this sequence at about 700 base pair or more, the sequencing quality will also drop. And the reason is that if you want to separate DNA fragments that are really long, the sensitivity is not that good, and that's why the quality drops at long fragments. The typical kind of error for Sanger sequencing is for a sequence that has a stretch of the same nucleotide, and basically they have the same color. And when you run the gel, you are trying to find a one base pair difference. And if you have the same color, it's very difficult for the detector to separate them. That's why most of the error is found in this kind of region. So it's called homopolymeric region. And based on this kind of, so remember that when you extract, when you try to sequence the DNA of an individual, you have 2 copies of their genome because humans are diploid. So you can actually read their genotype from this chromatogram. And if you look at this specific position, at this position, it is TT because there's only one signal, one color here. And this is only blue color that represents CC. But if you look closely at this position, it is a blend of red and blue. So it actually indicates heterozygote at this position. And this is personally, for my first research experience, I was doing this kind of sequencing experiment. And it was actually kind of interesting to see this kind of result once you do the sequencing, and then you visually go through those figures, try to identify genotype for your target SNP. And at that time, it was still in 2008, so Sanger sequencing was kind of the norm at that time. But right up to 2008, the next generation sequencing happened. That's the second generation sequencing. And Sanger sequencing is not used as often as before. And some other features in Sanger sequencing is that, so the sequencing we talk about start with a template, but it is very important to get high quality DNA template. And you want to have pure and also a large amount of DNA. And Sanger sequencing was developed in the 70s, but PCR was developed in the 80s. With PCR, now you can amplify your DNA template. You can get a very high quality template for DNA sequencing. So it makes Sanger sequencing more effective. The other major technology is called pair-end sequencing. And basically you can so usually when you you try to sequence a DNA fragment, you read from one end, and that's the sequence from for this place is called a read. So basically the sequence of nucleotide you read from the machine. Pair-end sequencing enable us to read the other strand of the same fragment. So you can get 2 reads from the same DNA fragment, and pair-end sequencing is very important for us to assemble genome. Another major technology is called shotgun sequencing. And basically shotgun sequencing is that for DNA is a long molecule. We won't be able to sequence one long DNA from the beginning. What we do is, because each read is only 800 base pair long, you won't be able to do that whole DNA fragment. And usually DNA fragment, the long DNA is fragmented into smaller sequences. And then each small fragment is go through the Sanger sequence. So you get the sequence for each fragment. So this is like a shotgun. You fragment that stretch of DNA into smaller fragments, and they sequence each fragment one by one. And then once you get, based on the sequence you get, you can, based on their overlap, to assemble them back into the genome. And for the Human Genomes Project, so that's the project that generates the human reference genome. It used the Sanger sequencing was the major sequencing technology. So this project was started in 1990s. So that's when Sanger sequencing becomes mature. And this project adopts a hierarchical shotgun strategy. Basically, it has multiple layers of shotgun sequencing. So we start with genomic DNA, very long DNA fragment. We fragment them into relatively large DNA fragments. So it's about 200 kb or so fairly long fragments. And those fragments are cloned into a back library. So it's basically bacteria vectors that we insert the human DNA fragments into that bacteria vector so that we can culture bacteria and then amplify that fragment. We can get a large number of those fragments for sequencing. And then for each fragment, you can apply another layer of shotgun sequencing, and then you get the sequencing reads. When you try to assemble, you go back, you assemble the back first, each back, and then based on each back sequence, you assemble back to the genome. And that is the strategy used in the Human Genomes Project. So for the Human Genomes Project, besides the sequencing, the small pieces of sequencing reads, they also need to use other information to help them assemble those reads back to the original human genome. Because after you do the sequencing, what you get is tiny fragments of sequencing reads. Each one is 800 base pair long, but the human genome is 3 billion base pair long. How do we piece those tiny pieces back to that huge genome? We can use this. So we can start with, this is the starting step. We look at the overlap between sequencing reads, kind of assemble them into relatively larger, we call cantig. Cantig is basically you assemble sequencing reads into a longer fragment of DNA. Now that you have the cantig, we need to use other information to put those, because we need to know where those cantig from. Where are they on the chromosome? And this kind of information is called contiguity information. And one of the features that make genome assembly challenging is repetitive sequence in the genome. And sometimes repetitive sequence, basically they are the same kind of sequence, and they happen many times in the genome. And so it will be, if you have a sequence that overlap with the repetitive sequence, then it could be assigned to many different places in the genome. So that's why we could not piece them together by themselves. We need to take advantage of some additional information independent of the sequencing. And that is genetic map and physical map. Physical map basically is based on the back library. When we try to construct back library, we know where those large fragments are from in the genome. We know the relative position of this back library. So once we assemble this back library, we can, based on the physical map, and then put them together. So physical map is one kind of information. The other kind of information is genetic map. Genetic map is a measure of genetic distance between some genetic marker, like single nucleotide polymorphism. And those genetic markers, so linkage mapping can identify genetic markers. And based on a genetic map, it can further help us assign cantig back to the genome. And tiling part. So this is just a minor concept. I'm not going to test you on this. This is just if we collect all the backs that by merge them, they can represent a specific genome. So we say that this set of back library form a tiling part. It was a concept used in genome assembly. And the other information, besides the genetic map and physical map, pair-end sequencing, this kind of information is obtained from sequencing. So genetic map and physical map are obtained independent of sequencing, but pair-end sequencing is obtained from sequencing. And so with pair-end sequencing, so for example, you imagine that we start with sequencing reads. We can assemble those sequencing reads into a consensus, and this is called a contig. We can get many contigs, but those contigs has no overlap, so we won't be able to assemble them based on overlap. What pair-end sequencing can do is that imagine that there is a DNA fragment, and that DNA fragment was sequenced. So we have two sequencing reads from that fragment, one from this contig, one from this contig. Based on that pair-end sequencing, we can link those two contig together. And once we link contig together, we call it a scaffold, just a concept. And one difference between contig and scaffold is that in contig, all nucleotides are known. In scaffold, sometimes you can have a gap. Based on pair-end sequencing, because when you do pair-end sequencing, we pre-select the size of DNA fragment. So we know that the pair-end, those two reads, the distance between them is about the size we selected. It's about the size of DNA fragment. So we can kind of estimate the size of that gap. So we can fill in unknown, like n, in those places. So scaffold usually has unknown or has missing data about nucleotide. And based on scaffold, so in this case, we create this kind of scaffold. And then we can further link scaffold to the genome using genetic markers or physical map markers. So that was first generation sequencing. Now we move on to the second generation sequencing. Second generation sequencing, also known as next generation sequencing, and it got that name at the very beginning. Now it turns out that it's already the previous generation sequencing. So one of the key features of the next generation sequencing is it is a massively parallel sequencing. Basically, it can sequence many DNA fragments at the same time. And the way so this is a general workflow in the second generation sequencing. The same as single sequencing, you start with genomic DNA, you fragment the DNA into smaller DNA fragments, and this is a unique process in the second generation sequencing is that you link some unique sequence to the end of those DNA fragments, so that the purpose of that link is that based on that link, you can bind those DNA fragments to a flow cell. So this is a flow cell. A flow cell is basically just a glass slide. This is a flow cell. On the flow cell, it has different channels. The channel is like a small container. You can have the sequencing mixture go through that channel. So the sequencing reaction will happen in that channel. So in those channels, there are many tiny wells, and in each well, it was pre-planted or embedded those oligonucleotides. So the oligonucleotide is designed so that they can pair with the adapted sequence in the target DNA. So once you apply the target DNA onto the flow cell, the DNA will base pair with the oligonucleotide already on the slide. So you kind of physically attach the DNA to a specific place. And then go through this process called bridge PCR. Basically, it's just a PCR amplification happen on a solid surface. And the purpose of the bridge PCR is that you start with one DNA fragment. after PCR, you amplify many fragments. And this is only the information from one well. And on each flow cell, there are many of those tiny wells. So you can imagine each well is one sequencing reaction. You can have many sequencing reactions happen at the same time.
Sorry if you already said this. What does each color represent in the native DNA?
Say that again?
Like, what does each color represent? It's like purple, then orange, then blue, then green, then is that the oligonucleotide?
So your question is, do the color represent anything?
Yeah.
Right. So here, the purple color here represents the adapter. It is an oligo sequence. We add it to the DNA. So we start with DNA fragment. That's our target DNA. We want to know the sequence of that target DNA. We add adapter sequence to the two ends of the DNA fragment. And the purpose of that adapter sequence is that they can base pair with the oligonucleotide on the slide. So the purple is the adapter sequence. And the color here is not really important here. It could represent different base pairs.
Okay.
Yeah, but the color here, for the purpose of this class, it doesn't represent. It's not important at all.
Okay.
You just need to know that this is a bridge PCR process, and bridge PCR is a key technological feature for the second generation sequencing. So once you get this cluster, okay, so bridge PCR is only one technology we can generate a large amount of template. An alternative to bridge PCR is called emulsion PCR, and basically it's a PCR happen in a droplet in the oil. And basically, you pre-design some microbits. Those microbits is like the glass slide we talk about. Like it's just like the flow cell. In this case, you use microbit, and those microbits pre-coded with some oligonucleotide. So similarly, they can capture DNA, target DNA fragment. And the idea is that each microbit only capture one DNA fragment. So when you do, and then later, each droplet, so this is a liquid droplet in a solution of oils. So each droplet is kind of like a tiny reactor. You can have a sequencing reaction happen in that droplet. So in each droplet, you first amplify the template DNA so that you have a large amount of targeted DNA template for sequencing reaction to happen. So those two methods, bridge PCR and emulsion PCR, are two approaches to amplify targeted DNA fragment. And bridge PCR is used in the Illumina Solexa sequencing technology, while emulsion PCR is used in Roche 454 technology. So those are two, basically Illumina and Solexa is one company. And then Roche and 454 is another company. And it has a complicated name. Solexa is the company, it was a small startup that first developed the sequencing technology. And later Solexa was acquired by Illumina. And then 454 was the startup, and then Roche acquired 454. And 454 was actually the first second generation sequencing technology available on the market. The thing is that right now the technology is already discontinued because it was outcompeted by Illumina Solexa. You don't need to remember that for the exam. And then, so the first feature is the technology to amplify DNA template. The other major new technology is the technology to read the sequence of the nucleotide. And the first new technology is called pyro sequencing. Basically, so this is the template, this is a primer, and you have DNTP try to incorporate into based on base pair to pair with the template. If this is the correct DNTP, once it is incorporated, it will release a pyrophosphate. And pyrophosphate can be used to generate ATP, and ATP can be used to trigger a fluorescence. So when a specific DNTP is incorporated, it will release fluorescence, so we can detect which DNTP is incorporated. So this is pyro sequencing, and pyro sequencing is used in the 454 technology. An alternative is called sequencing by synthesis with reversible dye terminator. It's a very long name. First, sequencing by synthesis is basically any sequencing technology that uses DNA polymerase to synthesize the complementary strand of the template DNA. So we have the template DNA, it's usually single-stranded. We use DNA polymerase and try to incorporate DNTP to synthesize a complementary strand, and in that process, we read which nucleotide is incorporated. So that's called sequencing by synthesis. And remember that Sanger sequencing is also sequencing by synthesis. Pyro sequencing is the same. And here, they emphasize sequencing by synthesis because they are in comparison with another technology I'm going to mention later. But the key success factor in this technology is the reversible dye terminator. So they are DNTP with specific modification. So they have the terminating and fluorescence group. So those two groups, the terminating group is reversible. So once that DNTP is incorporated, you could no longer elongate that DNA. It's the same as ddNTP we mentioned in Sanger sequencing. The difference here is that for ddNTP, it's not reversible. Once it's incorporated, you can no longer elongate that DNA. For this specific technology, once you incorporate it, you can read the signal, and once that signal is read, you can remove that terminating group so that you can restart the process again. So it's reversible terminator. And so this reversible terminator enable each time there's only one DNTP incorporated. And this is the technology used in the Illumina Celexa machine. The other technology, in comparison to synthesis by sequencing by synthesis, is the sequencing by ligation. So this is not rely on DNA polymerase. It's rely on DNA ligase. And so this is sequencing by synthesis. You have a template. Each time you incorporate one DNTP, one base at a time. For sequencing by ligation, is try to incorporate an oligonucleotide. It's no longer one nucleotide. And this technology actually is not that common right now. So I just put it here as a comparison. So this is the overall view of the Illumina Celexa sequencing technology, and this is the most successful technology in the market right now. So you start with genomic DNA, you fragment those DNA, apply the DNA to a flow cell, you went through amplification, and then at each place, you can go through this sequencing by synthesis with terminators one by one, and each round we can read which DNTP was incorporated, so you can read the sequence of the DNA. For the ROSE 454 sequencing technology, is a combination of emulsion PCR and pyro sequencing. So actually you need to, so these two pieces of information, you need to understand that. So 454 is the combination of the emulsion PCR and the parallel sequencing. I already talked about this in the previous slides. You capture the target DNA fragments by using microbits. So each droplet has one microbit carrying one target DNA fragment. You went through emulsion PCR, so you amplify that target DNA fragment. You then assign those microbits to different cells in a chip. And then in each cell, you can went through a parallel sequencing reaction to identify the sequence of that target fragment. So we talk about the parallelization feature, the new sequencing features, and another major feature of the second generation sequencing technology is that the sequencing result, the read from those machines is usually very short. It's only, when you first started, it was only 35 base pair. Now it improved to 25 base pair. But compared to Sanger sequencing of 800 base pair, this is short. And short read sequence has many problems, especially for if we have a repeat that have a very long, your read is not able to read through that repeat. So in that case, this read can assign to multiple places in the genome. So sequencing reads create a lot of challenge in genome assembly. And this sequencing technology has relatively high quality, but it is not as high as Sanger sequencing. And right now it's quite cost effective. It's less expensive than the Sanger sequencing. Later, I'm going to talk about the impact of Sanger sequencing, the second generation sequencing, what we can do with the second generation sequencing. But before that, any thoughts or any questions before we take a break? Questions about Sanger sequencing or second generation sequencing fragment? And so it's not very clear in this figure, but actually when we extract genomic DNA, we usually extract genomic DNA from a group of cells, from tissues, so it's many cells. Each cell has a copy of the genome. And each cell has 2 copies of the genome. So actually you have for example, for chromosome 1, you have many copies of chromosome 1. So for the fragmentation, there are several technologies can do fragmentation, and we don't care about the detail. We just know that there is some technology that can randomly fragment those DNA molecules. And the fragmentation pattern will be random for all the DNA for chromosome 1, for example. So the DNA fragment from different original DNA molecules, they have overlap. That's how we get the overlap from this overall DNA. And so the second generation sequencing technology, it kind of matured in 2007, 2008, and it was truly revolutionary. First, it significantly reduced the cost of sequencing. And remember that in the Human Genome Project, it takes about $3 billion to sequence the first human genome. But now if we many order of magnitude less than that. Right now it takes less than $1,000 to sequence an individual genome. And that's because of the the second generation sequencing technology. And the drop of the sequencing cost is much faster than Moore's law. You don't need to know what is Moore's law, but you just need to know that the sequencing cost dropped dramatically.
Do you know why it increased in 2016?
Here? Yeah. I don't know. Yeah, good observation.
Does the $1,000 sequencing include the analysis?
No,
Okay.
no way. Yeah. That's why bioinformatician is still a good career. Now we have a lot of data, but the company, they're going to give you the data, but what you do with that. And actually, right now in some company, they offer, for example, $400 whole genome sequencing. So it's way lower than $1,000. And the reason is that some company at the beginning, they have a very aggressive marketing strategy, and the approach is that they want to collect a lot of data. And usually if you get a low-cost sequencing package, usually you contribute your DNA to the database. So if they have a huge database, they can do a lot of things. So they kind of compensate their sequencing cost right now from their database. But anyway, the sequencing cost overall right now is very, very low. So with the second generation sequencing technology, you can do a lot of things. And the third thing is, similarly, you can use the second generation sequencing technology to do a de novo genome assembly. And remember that genome assembly is used to assemble the first genome for a specific species. And it reduces the cost for sure. The problem is that there's some problem with short-read sequencing, and the problem is that you could not resolve repetitive region. And the other thing is that although with sequencing technology, you can get pair-end information, but the problem is that for those pair-end fragments, it's usually restricted by how long the DNA fragment can get. And pair-end is able to provide information for specific distance. So if the two contigs is very far away, you won't be able to get a pair-end reads to link the two contigs together. So you need additional contiguity information like genetic maps and physical maps. We still need those in order to get a high-quality reference genome. But still, the current approach, the current strategy is usually to combine the second-generation sequencing technology with other technology, like the back sequencing. We want to continue to construct back. Still, we need to have genetic maps. And additionally, they're still based on the second-generation sequencing, but you can measure the 3D structure of DNA. Basically, for the DNA molecule, if the DNA for the DNA molecule from one chromosome, DNA they kind of folded. So the fragments they are from the same DNA molecule, they tend to contact with each other. So if you use a 3D genome sequencing, those sequences that contact with each other will be identified. So based on that information, we know that those two pieces are from the same chromosome. So it's still second-generation sequencing, but it provides additional information to link those pieces back to the genome. But second-generation sequencing by themselves is not sufficient to get a high-quality reference genome. The most significant application is actually in genome resequencing. Basically, genome resequencing is to sequence additional genomes in addition to the reference genome. And you can generate the sequencing reads like a very cost-effective method. And the benefit is that you already have the reference genome you can compare to, so you can map the sequencing reads to the reference genome and to identify genetic variants. So we already covered this in the previous lecture. And genome resequencing, so those are the three technologies I mentioned in the 1,000 Genomes Project: whole genome sequencing, whole exome sequencing, and targeted sequencing. They are actually all resequencing because you already have the reference genome. For whole genome sequencing, it's simple. You just extract DNA, you just sequencing everything in the DNA. For exome sequencing, you only sequence the exome, the 1% exome in the whole genome. The question is that, so we can, like for example, I collect some blood, from blood I can extract DNA, but those DNA is the whole genome. How do we fish out the fragments that correspond to the exon. And it was mainly done by, it's called capture. It's kind of like you design some probe. Because for human genome, we already know where are the exons. So we know the sequence for those exons. We can design oligonucleotide that match to that region. So it's like a bait. We can use that bait to fish out fragments that base pair with those oligonucleotides we have. So we can fish out those small regions correspond to exon. And then when we sequence those regions covering the exon, for targeted sequencing, similarly, you can use a probe-based hybridization approach to fish those target regions out. But if you have a small region, a region that is a few hundred base pair long, another approach is that you just do a PCR amplification and amplify that region. But PCR amplification is not really applicable for whole exome sequencing because there are too many places in the genome. And those are the major technology features you need to understand them. For whole genome sequencing, if you want to get high quality, usually you need to have a 30-fold coverage. Basically, you need to sequence every region on average 30 times. For this, you can sequence even more. So, because you have only targeted place, you can reduce the cost. And genome resequencing, because of genome resequencing, there are many large-scale projects trying to understand genetic variations in the human population, and also try to study the effect of genetic variation on human health. And we mentioned this project, the 1,000 Genomes Project, in last lecture. There's many other projects. For example, the whole exome sequencing project that generate whole exome sequencing for over 6,000 individuals. And this is a website, an online website that collects exomes and genomes. And right now there's about over 15,000 genomes available in this database. So there's a huge amount of genome already available because of the second generation sequencing data. There's many big projects going on. This precision medicine initiative, they try to do whole genome sequencing for another 100,000 individuals. And that's because of second generation sequencing. And so those are the two. So we talk about de novo genome assembly, genome resequencing. We can also use the second generation sequencing technology to sequence RNA, sequence the transcriptome. And imagine that you have the mRNA, isolate mRNA from the sample. You just need to convert your mRNA to cDNA. Once you get cDNA, it's the same procedure you do with DNA sequencing. And you similarly get the short sequencing reads, and you can map those reads back to the genome. And the one notable feature of RNA sequencing is that for some of the reads, because mRNA is only the exon, and exons are separated by intron in the genome. So for some of the reads, it could separate it very far away from each other. Basically, in between them, that is an exon. So when you do a read mapping for RNA-seq, the procedure is slightly different. But right now, it's a very mature technology already. So based on RNA sequencing, we can measure the expression level of specific gene. You imagine that there are two genes. One is highly expressed, one is has low expression level. For the one that has high expression level, basically you have many mRNA, many copies of that mRNA. So if you convert that into many copies of cDNA, that gene is going to have many sequencing reads. So when you map those sequencing reads back to the genome, the high expressed genes will have very high sequencing depth. A lot of sequencing reads come from this region. For other places that is not expressed or lowly expressed, the sequencing depth will be lower. So based on that difference in sequencing depth, you can kind of measure the expression level of each gene. So that's one major application of RNA-seq. The other application is that you can identify some transcript that are unknown previously. So that's RNA-seq. Ribosome profiling, similarly, it is sequencing mRNA, but you only sequence the fraction of so this is a mRNA, and those tiny circles represent ribosome. So those ribosome is trying to translate the mRNA into a protein sequence, amino acid sequence. So if this position is bound by a ribosome, you can apply some chemical so the ribosome will stop at that place. And then you just apply some enzyme to remove all those mRNA that is exposed. Only reserve the fragments that are protected by the ribosome. And then you sequence those DNA fragments, you can get to know where the ribosomes are on the mRNA. And usually, when you do ribosome sequencing, you also do a RNA-seq, so that from ribosome you can get to know how many mRNA is being translated. And then RNA-seq give you a background, how many transcript is out there. So you normalize RNA-seq result by ribosome result by RNA-seq result. It give you an idea of how active a specific gene is translated into protein. So ribosome profiling is another application. We can also do ChIP-seq. So ChIP-seq is similar to ribosome. Ribosome sequence the fragment that is protected by ribosome. We can sequence any DNA fragments that is protected by any protein. So ChIP-seq, chromatin immunoprecipitation, is a experimental approach to help you pull down DNA fragments that are protected by a specific protein. So this, for example, this is the DNA. Some of the DNA are protected, are bound by specific proteins. It could be transcription factor. And you can pull down those DNA and then only sequence those DNA. So you, by doing sequencing, you know where in the genome is bound by a specific protein. So that's ChIP-seq. And then metagenome and microbiome. We have a lot of microbes in the environment. We have a lot of microbes in our gut. As long as you can get DNA, we can sequence them. We can identify them. And for example, we can identify new species in some remote environment. For example, in the deep sea sediment, or in some in other weird places. As long as you can isolate DNA, you apply them to second generation sequencing, and then by comparing DNA to some available database, you can get to know if there's any new species in your sample. And now microbiome, so the microbe that live inside our body has been found to be very important for human health. And this kind of study was only available very recently because of the second generation sequencing technology. And before I go into the third generation sequencing technology, actually I want to mention the DNA microarray is not sequencing. It's just another technology to measure genotype, another technology to measure gene expression. So microarray is basically a solid surface. So this is a microarray, it's a solid surface, and on the surface there are many pre-designed oligonucleotides. We can pre-design those oligonucleotides so that, for example, if we are looking at a SNP, we have two alleles. We can design one of the oligonucleotide to match one allele, the other oligonucleotide to match the other allele. So when we apply the DNA of a participant to this microarray, we can look at which position is bind to the target DNA. So we can identify what kind of genotype that individual carries. Similarly, we can design oligonucleotide that base pair with specific gene, and then we can apply mRNA samples to a microarray. If a gene is highly expressed, a lot of these probes will be bound to the target mRNA. With that, based on the amount of signal from the microarray, we can also measure the expression level of specific gene. So microarray is a very efficient approach to genotype null position or to measure the expression of pre-selected genes. It's only designed for null places. And microarray, because of microarray, that we actually, before the 1,000 Genomes Project, there's other large-scale project that was designed to study human genetic variation. And the most famous one is the HelpMap project. HelpMap project using genotyping array to study common variations in a large number, a few populations. And they study 3 million SNPs. So those SNPs are pre-selected. So they won't be able to know any new mutations in the population. And most of the genome-wide association study that try to find association between genetic markers and a specific disease is based on genotyping array. Because genotyping array is much, much cheaper than sequencing. It's even cheaper than targeted sequencing. Okay, so and then we move on to the third generation sequencing, the current generation sequencing technology. And the major feature is that it's amplification free. Both single sequencing and the second generation sequencing, you need to amplify your template. The amplification can create a lot of problems. It introduces error, it creates some context-specific bias, and you also lose information, for example, methylation. So the third generation sequencing bypass the amplification step. So we are going to introduce two major sequencing technology. The first one is called paired biosequencing. So the paired biosequencing has this is a unique technology. It has a physical structure. It's called zero mode wave guide. It's just a real name. It's just remember that it's a physical structure that confines the template, basically the single molecule DNA in this place, and also other reagents in this place. So once a specific nucleotide is incorporated into that DNA molecule, it can emit a specific signal. And then based on that signal, we can identify which nucleotide is incorporated. So this kind of technology, the unique advantage is that it has very, very long read length. The longest could be 100 kb. But the problem is that it has low throughput and it has very high error rate, much higher than the second generation sequencing technology. The good thing is that the error is randomly distributed. It suggests that potentially you can correct those errors by sequencing the same place multiple times, so that it's unlikely for error to happen at that same place. Nano sequencing, basically it has a nanopore, a nanoscale pore channel. Once you pull the DNA molecule through that channel, it can trigger a change in the signal, so you can reach the signal is specific to each nucleic acid, specific to each nucleotide. So once you pull that DNA fragment through that channel, you can read the which nucleotide is incorporated. So this technology similarly has very long read length. The error rate is similarly high. One unique feature of this technology is that the device is very small, so it has very high portability. And these two technologies are still in development, and they're still in the process of improvement. But with the long read sequencing, you can do quite some new things compared to the second generation sequencing. For example, identifying structural variation, they are very long. Actually, you need to use long read sequencing. That's it. Thank you.